So we learned about Mendelian patterns of inheritance, the ones that follow Mendel's patterns. But what about those that are a little more complicated or that like to follow their own path? We're going to learn about those non-Mendelian inheritance patterns now. So Mendel said that the dominant allele always overpowered the recessive allele. The traits are always inherited separately from other traits, and that when you have one dominant allele, it's going to always show up. But we're finding that in real life, that's not always the case. It gets a little bit more gray as we move forward. So while his laws are still true, we have to add on a deeper understanding now. And remember that Mendel did all of this work without any knowledge of DNA. He was prior to the 1850s, so this was really before we even knew what was inside of the nucleus. So he was pretty advanced for his time. Once we really came to understand DNA, we found that all of his rules still held true, but it was just a little bit more complicated than the basic ones that he laid out for us. So we're going to learn about incomplete dominance, codominance, and many other unique inheritance patterns today. So there's a couple terms to know. The first is wild type. This is what scientists use to describe a gene that exists in its normal, natural environment. This is what you'd find naturally occurring. A mutant is just any allele of the gene that's not typical or not wild type, or that we've induced to be a little bit mutated, essentially. Now with incomplete dominance, the first one that we're going to look at, neither allele for the gene is completely dominant. So you're going to see a blend of the traits, just like in this picture here. You have the light bulb and the swirl ice cream cone, and you produce the swirly light bulb as the offspring. So the two examples in animals and plants are the 4 o'clock flowers and the mouse fur color. So let's take a look at those. 4 o'clock flowers have a red phenotype and a white phenotype. That's true breeding. But when we cross these two true breeding plants together, the F1, or the first generation, is going to be pink. And pink is just, as you know, a blend between white and red. So this is a case of incomplete dominance. Neither the red nor the white was completely dominant, so you see a blending of the two traits. In these cases, anything that's heterozygous is going to have a blend of that trait. And there's a couple ways that we can write this. You can represent the red flowers as R's, or you can represent the flowers with an F, and then you just have to put that they've got the red color on them or the white color on them as a superscript. The same thing happens with fur color in mice. You have black fur, which is homozygous dominant. You have white fur, which is also homozygous dominant, and then you have an incomplete dominance happening when you get the heterozygous mix. You get the gray-furred mouse instead of just pure white or pure black. So let's take a look at this example here. A mother has straight hair and a father has curly hair. Create a Punnett square to show how their daughter could have curly hair. So if we create this Punnett square here, so we have a mother who has straight hair, and in this case, straight hair and curly hair are going to be the dominant traits, and the wavy hair is going to be that heterozygous trait that we see an incomplete dominance with. So the mother has straight hair, and I'm going to represent that with S's, and the father has curly hair. I'm going to represent that with C's. So when we do the cross of our Punnett square, we're going to see this mix here. All of the children are going to receive the same genotypes. they're going to be mixed. They're going to show that heterozygous incomplete dominance. So what's the probability that their child will have curly hair, that they're going to look just like the dad here? Well, the probability is 0%. All of the children are going to have the incomplete dominant wavy hair. None of them are going to have curly hair. So let's look at a different example, a very cute example. Golden retrievers can be a blend of colors ranging from dark red to golden or very, very light golden, almost white. And we can use a Punnett square to predict possible phenotypes. In particular, what could the possible phenotypes be if we mix a dark red golden dog with a golden dog? And I need a little bit bigger of a Punnett square than that to do this. So we are mixing a dark golden dog with a regular golden dog. So we're mixing one on this end with one in the middle. So in this case, I'm going to have hair color that is dark for one, and hair color that's going to be in the middle, an incomplete dominant. It's not going to be dark and it's not going to be light. So when I do this cross, I can already see that I'm going to get different results from the previous example. In this case, I'm going to have 2 out of the 4 offspring with a dark coat, and 2 out of 4 of the offspring are going to have a golden coat, that incomplete dominance color, the one in the middle here. So what are the possible phenotypes that can occur? Well, the phenotypes are dark golden and golden. Those would be the phenotypes that I would get. If I wanted to know the genotypes, you would have to write whatever we put inside the Punnett square. So I want you to do this example on your own. Cross the following 2 snakes. You have a wild type that's represented by W, and what's called the super jungle type represented by J. So you have super jungle on one end, wild type on the other, and then you have a heterozygous that's called just regular jungle. I want to know what are the genotypic and the phenotypic ratios crossing these 2 snakes here, a heterozygous and a homozygous. So let's go over what you did. In my Punnett square, I'm going to be crossing a heterozygous jungle and a super jungle python. And when I do this Punnett square, this is what I get as a result. I get super jungles and I get regular jungles. So 50%, 2 out of 4 of them, are going to be those jungle characteristics, and 50% are going to be, I'm sorry, I messed that up, super jungle, and the other 50% are jungle. So what's my genotypic ratio? Well, it's going to be a 1-to-1 ratio of that super jungle to jungle. This will be for genotype. And then my phenotypic ratio is also going to be 1-to-1 because I'm going to have 2 super jungles and 2 regular jungles. So when I reduce that number, it's 1-to-1. So I'm going to have super jungle and just regular jungle. And that's my phenotypic ratio. The next particular inheritance pattern that we're going to learn about is codominance. This is where neither allele for the gene is dominant. Both are equally expressed and both show up. So I'm not going to see a blend, I'm going to see both of the dominant alleles. So just some silly examples. This is not how it works in real life. Crossing the black bear with the white bear would end up with black and white bears. crossing a beaver with a duck gets you a platypus. Not in real life, as I said, but it's funny to joke. So the first example are these white and black speckled chickens. So a full black chicken is going to carry the black dominant allele. The white chicken carries the white dominant allele. And when you have the heterozygous chicken, you end up with the speckled colors like you see in these two pictures. They have a mix of black and white feathers. We also see this in our blood cells with sickle cell anemia that we've already talked about. You can have healthy, normal blood cells, or you can have that sickle-shaped blood cell, the diseased blood cell. Or when you are heterozygous, you can get that mixed heterozygous characteristics and get a mix of the blood. So let's do this example together. A roan cow is produced when a red and white cow breed, and they'll experience codominant inheritance. So this is the roan cow at the bottom, and we're going to cross the red and the white cow to get that situation. So what would be the phenotypic ratio when a red cow is crossed with a roan cow? So in this case, my cow that's white is going to have this particular genotype. The red cow will have this one, and the roan cow is going to be the mix of the two. So let's do this cross now that we have our genotype straight. The red cow is going to be crossed with the roan cow. And when I do that cross, I end up with this particular mix of my alleles. I'm going to end up with red cows and roan cows only. So what's my phenotypic ratio? Well, it's going to be 1 to 1 again. 1 roan for every 1 red. So a snake breeder wants to know how to create an interesting mix between a wild type and an albino python, like in the picture shown, where you see normal bandings of color mixed with albino bandings of color. So you're going to create the Punnett square to show the breeder what parents you're going to need to get this heterozygous mix. So there's a couple different ways we can do this one. You can start by just filling out a Punnett square with the mix that you want to create. So we can say this is a python and we want to mix normal with albino. So that means I'm going to need to put that inside my Punnett square. How would I get to this particular case? Well, I would need a normal python allele there and an albino allele python here. And if I wanted to make sure that I definitely got every single possible python with that cool mix, I would want to fill in every single square with the mix that I'm looking for, that heterozygous mix. And when I do that, I find that I'm breeding a completely normal python with a completely albino python to make sure that I get every single offspring with that mix. So what would be the genotypic ratio for a cross between an albino peacock and a half albino peacock? So we're going to have a Punnett square, and this is how I'm going to set it up. The P is going to represent peacock, and I can have a normal peacock, or I'm going to have the albino peacock, or I can have that heterozygous mix like that awesome picture that we see right here. So I'm crossing an albino peacock, which is going to have the 2A alleles, with this half albino heterozygous one. So it's going to be a normal allele and an albino allele. And we're going to do that cross. And when I do that cross, these are my possible results. So I want to know the genotypic ratio. Well, the genotypic ratio, my genotypes are going to be a heterozygous compared to homozygous, and I'm going to have 2 to 2 or 1 to 1. That would be my ratio. Another really cool example of codominance is our human blood type system, our ABO blood types. So humans have 4 major blood types that you should know of. There's AB, AB, and O. Our blood cells have proteins on their surface called antigens, and the type of protein on the surface is going to determine whether or not we have that A, the B, the AB, or the O blood. And then we make antibodies. These are special proteins that can recognize those surface proteins, those surface antigens of blood that's not ours. That way, if somebody else's blood gets into our body, we can attack it and keep it from hurting us. So type A blood, this group right here, has A proteins represented by the little circles on its surface. And so they will make anti-B antibodies. We don't want any B blood getting in the A blood body. Type B blood produces these diamond-shaped, this is just an example, it's not really how they look inside the cell, the diamond-shaped B antigens, and they make anti-A antibodies. AB blood is our codominant situation. I'm going to end up with A and B antigens on the surface of this blood. And since I have both antigens, I want to make sure that I make no antibodies. Otherwise, my blood is going to attack itself. So in this case, we have no antibodies. And the last group is O blood, where I have no proteins on the surface. So I make antibodies against everything because every other type of blood is going to be different than my blood. So this shows codominance between the A and the B forming that AB blood. I can have one phenotype or the other dominant phenotype, or I can have that heterozygous mix of both dominant phenotypes. We can see this especially when we look at the genotypes of our blood type. I can have 2 A alleles from a parent, or I can have an A from one and an O from the other. And we see that this always makes A blood. So the O is recessive to the A allele. Same thing happens with B blood. In order to see that O blood, I have to have 2 O alleles, 2 recessive alleles. So we can write it in either way here, using the A's and the B's, or we can use an I with an A, a B, or an O. So let's do an example problem using blood type. We're going to take a mom who has heterozygous type B blood. Heterozygous means different, so that means she's going to have a B allele and an O allele, and a dad with heterozygous A blood. So heterozygous means different, so he's going to have an A and an O. So let's do this cross. AO and BO. And these are the possible variations that we're going to end up with. What possible blood types, so this is the phenotype that we're talking about, what's the phenotype that their children could end up having? Well, you can have a kid with AB blood, with B blood, type A blood, and type O blood. So this is one of the cool mixes where you can end up with kids with every single type of blood. Blood also has a second factor with it called the RH factor. And this is where we get that positive or negative. Are you positive for the RH factor or negative for it? It's a lot more complicated than that, and we're really only looking at one of many different RH proteins on the surface of our blood, but it's the next most common one that we know. So you can say I'm O positive or I'm O negative. And this causes a really fascinating scenario when you're talking about mothers who are pregnant. If a mom is Rh negative, she's going to make anti-Rh antibodies. She does not want any Rh positive blood inside her body. So if the dad is Rh positive, the baby could possibly be Rh positive. When that happens, the mom's antibodies are going to see the baby as foreign, and it's actually going to attack the baby's blood. Will create the antibodies to attack that baby, and you can end up with a miscarriage. Now, good thing is we've discovered this, and so the mom can actually take a special medication while she's pregnant, and it'll reduce the likelihood of her having a miscarriage. Another thing is usually the first birth is normal, and that's when you build up the antibodies if you're that if you're that Rh negative mom, and it's the second time you try to have a child that you're going to end up with the miscarriage. It doesn't always happen that way, but that is what we seem to find is the most common. Now there's another way that we can inherit traits, and that's through polygenic traits. Poly means many, and genic refers to genes. So this literally means many genes for one trait. That's like our skin color or our eye color. These are incredibly complex. We have so much variation and so many possible shades of skin color and eye color because we have many genes controlling that one particular trait. So an example of a disease or a syndrome that you get as a result of one of these polygenic traits is Waardenburg syndrome. And this is where all of these genes have mutations in them. It occurs in about 1 in 50,000 people. So you'll end up with a special facial structure. You'll end up with often having a stripe of dark or of white hair. You might have eye problems and hearing problems. We see this a lot in cats and dogs and ferrets that are white. They'll often be blind and deaf because of the way that those genes are linked together and interact. If you have an all-white animal, there's a higher risk that they are also blind and deaf because those genes are linked. So these linked traits are actually just genes that are very, very close together on the chromosome. So you can see that B and C are very close together, but A is far apart. So if we were to have a crossing over event, like what happens in meiosis 1, it's more likely that these B and C's are going to stay together and they're going to get separated from the A because the A is so much farther away. So we might cross over here and switch this information, but we don't often cross over up here and switch all 3 genes. So it's likely that B and C are linked and A is not. And we can end up creating these really cool large Punnett squares to look at these particular linked traits. It means that the letters will always come together. Now this is using A and B, but if we were to compare it to this example here, you would end up with B's and C's instead of A's and B's. Another interesting thing that affects your genetics is something called epigenetics. This is where the environment influences your genes. Depending on what environment you're in can actually make your DNA act differently. So maybe we'll make more protein because of the environment or less protein because of the environment. It doesn't literally go in and change the order of all of your bases. It just makes your genes act differently. So I can turn off a gene or turn it on, or make an on gene be even more on and more excited and producing more protein or something like that. A cool example of this type of epigenetic control is Siamese cats. They have really cool coloration patterns. They have dark tails, dark ears, dark paws, and dark faces, while the rest of their body has a lighter fur color. What do all of these areas that are dark have in common? Well, they tend to be colder areas of the body. When you look at your core, it tends to be a lot warmer than your fingers. Your nose tends to get a lot colder. Your ears get colder before your stomach gets colder. So this is the case of your genes being temperature controlled. Wherever they have a colder temperature, they're going to have darker fur color. Another example that's linked to temperature happens in alligators and some reptiles. You'll find that when they make their nests, they'll have a bunch of eggs all around, and the temperature is actually going to determine which ones are male and which ones are female. All of the ones on the outside are going to be colder than the ones on the inside. So you're going to see one sex on the outside and a completely different one on the inside. Kind of cool. Well, that's all I have for you guys today. If you have questions, just message me and let me know.